All right, today I'll be talking about the Lac Operon. This will be a quick review of the experimental history that we learned in Biochem 242, and hopefully it makes sense. So, our story begins with some E. coli cultures, as they usually do, and in this particular culture, I'll draw it here, we had a lot of glucose and lactose in the medium. And we saw this growth curve. And okay, this first part makes a lot of sense. It's exponential, but what doesn't make a lot of sense, or at least is interesting, is this little lag phase between the use of glucose and lactose. Now, this was very interesting because if these two sugars were metabolized in the same way or in conjunction with each other, this curve should look very homogeneous and it should be pretty continuous. But we do see a little lag phase in the middle. And so this implies that there's some sort of switch that was turned on after all the glucose was used up. And obviously, a big question for this would be, how is this regulated? How do we regulate the switch, and how does lactose play into it? How does glucose play into it? And so the very first thing that we want to see is how we came to a model of the operon. And the operon is basically just a series of genes that are all connected, that all get transcribed at once in the same RNA. And this was a model that was first proposed by the Lac Operon. And here's the basis for this model. Of course, we have beta-galactosidase. I'll write that here. Beta-galactosidase. And this just breaks down lactose into glucose and galactose. That's what beta-galactosidase does. And scientists found mutants that could not grow on lactose despite having this enzyme that catalyzes or allows the metabolism of lactose, right? And so this seemed kind of odd because if you have the enzyme, presumably you should be able to metabolize the sugar. But they found that these mutants couldn't actually bring lactose into the cell. And this, therefore, was deemed galactoside permease, or we can also just call it lactose permease, and this would be a transporter protein that could bring in lactose into the cell. So now we have another protein called lactose permease. And obviously, and lastly, we also have another one that's called galactoside. Galactoside. That should be like that. Transacetylase. And the function of this is still unknown. The function is known, but it's still unknown as to the significance of this. But I'll just call it transacetylase here. And it found that all three of these proteins were all coupled upon addition of an inducer. So an inducer in this case could be lactose, right? Because lactose ultimately produces allolactose, and allolactose drives transcription of these genes for reasons unbeknownst to these scientists, right? But IPTG, which is an analog, is not metabolized in the cell, contrary to allolactose, which is metabolized. So IPTG was used as a gold standard in these experiments. More about that later, but right now just know that with the inducer, all three of these proteins were encoded for. And this suggests some sort of structure, genetic structure, where these genes are really close together with one promoter for RNA polymerase to bind, and it transcribes these genes all in conjunction with each other. And these genes we'll call LAC-Z, LAC-Y, and LAC-A for beta-galactosidase, lactose permease, and transacetylase. So this was sort of the proposition of the model. But obviously, the inducer has to play into here somehow, right? There has to be some sort of regulatory elements in this model. And so that's what scientists tried to investigate next. And this was where the proposition of a lac repressor came in, because they found that there are some mutants that always express these three proteins despite whatever their environment might have, right? Even if there's inducer, no inducer, they always secreted these three proteins, or expressed these three proteins. And these were deemed I- mutants, okay? And I'll denote I plus here as wild type mutants, wild type bacteria that just did it normally. When there's inducer, they express these proteins. When there's no inducer, they're chilling. Okay, and this is a very interesting thing they found about this genetic mutation, because let's say I minus is red and I plus is green. When they have two green, right, two wild type, everything's great. Things are going amazing. When they have two red, as expected, there was constitutive expression of these three proteins. However, as long as you have one gene in here that is wild type, we rescued. Sorry, these should not be Z. This should be I minus and I plus. These genes, just addition of a wild type gene, rescued the mutant phenotype. And this also went for when there was two mutant genes, but on a separate plasmid, the transgene containing the wild type I plus gene also rescued the phenotype. Now, this suggests that this gene encodes for some sort of repressor, some sort of protein, all right, that could then bind to the main chromosome or the other strand and regulate expression. And this is what we call a a trans-acting factor. Because it can affect regulate the expression of genes not physically linked to it, right? This could be a transgene, it could be the other DNA strand, and it will be perfectly fine. Now, next up is what we call the operator. And the basis for this was because these scientists also found mutants that were also, constitutively expressing these three proteins. We'll call them OC and O+. And you might be wondering, why are they different? How do they know they're different? Well, when they have one OC and one O+, they saw still constitutive expression. And so it's not a transgene in that case, right? And so this was very interesting because this is a different property. It has a different property than the first mutant we saw. And so this suggests that this is a whole different gene, right? Now, scientists also ran experiments with partial diploids containing different variations of this OC mutant, and as well as a Z minus mutant. And Z minus, this is just non-functional beta galactosidase. And so what they found was, okay, when we have OC and Z minus, I'll draw that here. Let's say OC. It's like that. O plus. And so we had Z minus and Z plus. In this mutant, in this bacteria, this functioned perfectly fine. With inducer, it produced functional beta-galactosidase, and without inducer, nothing happened. It didn't metabolize lactose or anything. And so this suggests, well, for one, this mutant, right? It's the constitutive mutant, and so it would theoretically constitutively express the Z minus gene. And Z minus, remember, is the non-functional beta-galactosidase. And so that's fine. You can keep on producing non-functional beta-galactosidase. No one really cares because it's not working. Whereas this one worked perfectly fine, right? So this was one hint that this sort of gene wasn't really a gene at all, but more so like a regulatory region for subsequent downstream genes. Now, this was confirmed with the second partial diploid, where we had also, like, we had OC, right? O plus, but this time we had Z plus and Z minus. Now, this constitutive one would theoretically just keep on transcribing the Z plus constitutively, and the other one would abide by the same rules that of like has to have the inducer in order to transcribe, right? But it doesn't really matter because it was a Z minus gene, and Z minus, remember, encodes for the non-functional galactosidase. And what they saw in this strand was constitutive expression of galactosidase. So this kind of just confirmed that, okay, what we have is a repressor, right? A repressor will bind to the genes to repress their action, without the presence of inducer, that is. And we also have an operator sequence where presumably this Lac repressor would bind, right? And so now we're starting to fill the gaps, because now we know there's the operon, the promoter, but it's a bit more complicated than that. It seems like we also have some sort of operator sequence, and we also have a separate gene that encodes for a repressor protein that I guess presumably binds to the operator, right? That's sort of the line of thinking. And so this is the model thus far. We would have our promoter sequence, right? This is where our RNA polymerase would bind. RNA polymerase. We would have 3 genes, right? Lac Z, Lac Y, oh, that's a horrible Y, and Lac A. Remember, this is beta-galactosidase, this is permease, and this is acetylase. But we also have some sort of operator that has to be intact, because when we had a constitutive version, right, which probably just means a few nucleotides were mutated and didn't allow some sort of binding, right? If this is mutated, then we get constitutive expression of these 3 genes, meaning that this operator probably plays some sort of role in RNA polymerase binding, right? That's kind of the only way you can regulate transcription in this case. And we also have a separate Lac I gene. And this Lac I gene, it could be coupled with these 3, or it could not be, not too sure. Though it probably isn't, right? Because it did seem like the constitutive operator had much effect on that. This encodes for a repressor, which presumably binds to, the operator or maybe the promoter. We're not too sure, but probably the operator, because that's when we lost regulatory ability. So presumably this repressor binds to the operator. This is our model so far. And this is... And now, we just want to understand a bit more about how this repressing works exactly. Does it actually bind to the operator? How does it, do what it does? So first, they want to isolate this repressor. This makes a lot of sense. And remember when we brought up IPTG? This is the analog of allolactose, which is the natural inducer in bacteria. And so, this probably would bind to the repressing protein, right? Because, if the inducer is there, it probably binds to the repressor protein and, something happens. And so they also found a mutant Lac I plus mutant, what was called Lac I plus, that was inducible at lower levels. So that just means probably has like stronger binding, right? So, they use chromatography on the cell lysate of that mutant, and they tried to filter out this protein. And then they did a radioactivity assay. And the way they did this was they had big beakers and a dialysis bag. And in this dialysis bag, they had what, presumably would be the repressor protein. Obviously, they didn't know how big the protein was. So they had to run a bunch of, size exclusion chromatography methods and test each fraction on its own, right? Because they just didn't know how big this was. And in this water, they had radioactive IPTG. Radioactive IPTG. And IPTG can freely dissolve into this dialysis bag. However, obviously the protein is too big, so it can't diffuse out. And so if this does bind, right? If it binds to here, then this effectively would increase the concentration of IPTG in the bag. And since this is radioactive, we could test for this radioactivity. And that's exactly what they found. They found that there was a higher level of radioactivity within Lac I plus mutants compared with their control. And their control, right, also would have these proteins, as well as the IPTG. But this would be the I minus mutants. Remember from last time, this is the non-functional repressor. And they found that this, the radioactivity inside the bag was not really, wasn't meaningfully different from the outside. But this radioactivity, on the other hand, was. And so this kind of confirmed that, okay, IPTG does in fact bind to the Lac repressor. But another thing is we want to see the binding affinity to DNA of the Lac repressor, right? Because, okay, this protein binds the Lac repressor, amazing. But we need to make the next step, which would be the repressor binding to the DNA. And so the way they did this is they had the repressor, obviously, from the Lac I plus. They had radioactive DNA sequences from the Lac Operon, and they also had a nitrocellulose filter. And nitrocellulose filter, basically what this does, it lets DNA pass through, right? You can see through this example in this a little figure. The DNA can pass through perfectly fine, but protein can't. And so if we just look at these experiments real quick, actually, let's say the radioactive is labeled radioactive, the DNA. Then in part A, right, this would have radioactivity. In part B, this no radioactivity, right? And in part C, up here would have the radioactivity. So that would give hints as to how much DNA was somewhere, right? It was just a proxy for DNA concentration and therefore binding affinity, right? And so in further experiments, they kept the number of DNA constant, right? So they had a certain number of radioactively labeled DNA, but then they varied the concentration of repressor and then later on inducer. And they would measure the radioactivity on this nitrocellulose filter because anything that got stuck up there, right, would be proteins that were, and if it was radioactively labeled, that protein would be, bound with the radioactive DNA. And so this obviously serves as a proxy for, DNA affinity. And these are the results they found. So down here in green, you can see our negative control, because they just straight up put no operator in here. So this is great. There was very little binding, maybe just some transient binding to random DNA sequences, but nothing substantial. Now you can see we're increasing repressor concentration, by the way, along the x-axis. And on the y-axis, we just have a measure of how much radioactivity was seen on the nitrocellulose filter. Now we see that for the wild-type operator, we have a very fast increase in the radioactivity measured as you increase repressor concentration. And this is very suggestive of repressor binding to this O plus sequence, and it seems to be very strong. Now, on the contrary, this OC mutant, remember, this was a constitutive one that always constitutively expressed the three genes, presumably because it didn't bind to the repressor, had much lower affinity. By the way, ignore this top part when this goes above. This is likely due to experimental error back then. They weren't, the preciseness of experiments weren't extremely thorough. And so this was likely just experimental error. But the point is, you can see the difference in binding affinities between the constitutive and the wild type operators. So this suggested that indeed the repressor did bind to the operator. So this is the operator, and this is the repressor. Now, we should look at also the inducer, right? If we add inducer, um, this repressor protein should come off to allow for transcription. And that's exactly what we see. When we add IPTG down here, you can see that there is very little DNA affinity binding. This suggests that the repressor bound to IPTG has very low DNA affinity compared to its non-bound IPTG counterpart, as you can see in the graph of the red. Much higher affinity when there is no inducer. And this makes sense, so this suggests that once IPTG binds to this repressor, some sort of change happens to where the repressor is now released from the DNA. That's sort of, what the experiment is showing. And so, obviously the next step now is how exactly does the Lac repressor block transcription, right? Because transcription is involved with RNA polymerase, and we now know that upon binding to IPTG, this repressor releases, but we still haven't figured out when it's not bound to IPTG and it's chilling on the DNA, how is that actually affecting RNA polymerase binding? We have two possibilities, actually. I'll list them here. Number one is that this repressor will be here. Let's say the repressor is here. And then we have RNA polymerase binding here, but it just doesn't let it go. All right, so we can't end up going, but RNA polymerase is still there. Okay, that's option number one. The other option is that here's our repressor, and this blocks RNA polymerase from binding in the first place. This is the other option. So these are the two models that we would be examining. And I would like to start off with just a misleading in vitro experiment that they found. Because what they found was, okay, this is called runoff transcription. So basically you have a little glass tube, and you add your DNA of interest, and same with your RNA polymerase. So in here you have your DNA, you have your RNA polymerase, and then you also add, you can, this experiment, you either added a Lac repressor or you didn't. And then you would add, let's say you added repressor, okay? You add a repressor. So those guys would mingle, and then you guys add, and then they would add heparin. And heparin basically prevents binding of any closed RNA polymerase complexes, and boots them off, but it doesn't affect the open complexes of RNA polymerase. And open complexes is when it actually fully binds and really gets in there, right? The closed complex is pretty pathetic. It hasn't opened the DNA yet, suggesting that it's actually not that well binding, right? And so that's basically what heparin does. And then you would add some labeled nucleotides, as well as if you want to add inducer as well. That's also another option. And so this would allow any bound polymerases to transcribe, and then you would run this result on a gel. And on the right, you can see the results. And the first lane would be a positive control, right? The positive control, because this is basically what we should see. They didn't add any repressor, and obviously if there's no repressor, you don't have to IPTG doesn't really make a difference, right? And so if you don't have any repressor, then the RNA polymerase should be able to bind and bind very well because nothing's stopping it. So when you add heparin, nothing really changes, right? It's not strong enough to yeet them out. And then when you add your nucleotides, there's transcription, and we see a big band up here, which is the transcribes, transcripts of beta galactosidase, galactoside permease, translitase, And then we had in lane 2 a negative control. And this is when they had the repressor and no inducer, right? So presumably the repressor will bind to the DNA, and this will prevent RNA polymerase from binding, and or if it does bind, right? We don't know if it binds or not, because after you add heparin, some of them will go off, some of them will stay on, maybe. Because we don't know. That's what we're trying to figure out, right? We're trying to figure out if RNA polymerase can even bind in the first place if repressor is here. But if it can't, or if it can, it doesn't really matter because you're not adding any IPTG, so the repressor is not going anywhere, and we know repressor works, right? We know repressor is not going to let transcription happen. And so this was just as a negative control to make sure everything's working. Okay, great, there's no transcripts. Maybe like a very light band because, maybe the repressor fell off at one point, right? But overall, this is not substantial at all. So that's great. And the real experimental lane is when you add lac repressor and IPTG. Because what happens is you have your lac repressor on your DNA, or I'll just write this out here. You have your DNA, and let's say you put your lac repressor in here, right? And you have your RNA polymerases just chilling out here. Now, we don't know if these guys are actually binding or not, or if they can't bind at all, right? So you add heparin, and that would remove all of the closed RNA polymerase complexes, right? And you wash those out. And so, these guys could either be bound perfectly great, or they could be out. And then we add both IPTG and nucleotides. So then when we add IPTG, these repressors would fall off, right? And any bound RNA polymerases would just go. And that's actually what we saw. We saw in this experiment that, yes, indeed, some RNA polymerases remain bounded, and quite a bit of them too, because these bands are quite thick, right? Meaning that, okay, I guess the RNA polymerase can bind even in presence of lac repressor. But this experiment was flawed, and this was mostly because it's conducted in vitro, and so the relative concentrations of these reagents aren't particularly representative of what's actually happening in a cell. But you do have to know this experiment because it was still one of the important hallmarks. However, it was the wrong finding because, well, I'll present the correct finding, is they used chromatin immunoprecipitation to actually figure it out in vivo. And the way it works, so you have your cells and you treat it with formaldehyde to basically form covalent bonds between any proteins and DNA if they are bound. And then you break the DNAs into pieces. You can see on the right here, you break DNA into pieces and you immunoprecipitate a certain protein of interest. In our case, it would be RNA polymerase. And then you can see, if it's there, right? You can break the cross links by increasing temperature, and then use PCR to see what that DNA strand is, right? And so the experiments they did was, first they had the cell, we lyse the cell and we get the solution, right? And then they either add IPTG or they don't add IPTG. And when they did add IPTG, they found that, okay, RNA polymerase did precipitate. This makes sense because you add IPTG, there's no repressor. Repressors are floating around, and so RNA polymerase should be able to bind anyway. And so when you cross-link these and you break it apart and you run it on the gel, you should get the transcripts. You should get the DNA that the RNA polymerase was bound to, because, nothing's stopping it. Now, this is the key, because when they did not add IPTG, meaning that the repressor was bound on the DNA, there was no RNA precipitation. Now, this is suggestive that if the repressor is bound, then RNA polymerase can't bind on. Otherwise, we would have seen RNA precipitation, right? Because if we didn't see any, right, that means this guy wasn't even able to stay on and be covalently cross-linked because lac repressor was preventing it, right? That's the only explanation, because once you immunoprecipitate it, we should have seen DNA on the gel once you run it from RNA polymerase, but we didn't. And so this confirmed that there was no RNA polymerase binding when the repressor is on the DNA. Whew. Okay, so that's solved. So now we'll just go over a quick review of what we know right now. So we know that the operon has 3 genes, right? 3 genes, 1 promoter and one operator, right? So we have a promoter sequence here. We have our lac Z, our lac Y, our lac A, right? We have our operator. We have our promoter. And we also have, some gene downstream that's for a repressor that then binds to the operator to regulate expression. And now we know that in the presence of IPTG, IPTG will bind to the repressor and release it from the operator, allowing RNA polymerase to transcribe. Now, without the inducer, this repressor stays on the operator and prevents RNA polymerase from binding in the first place. So that's what we know so far. Great. Our model's becoming a lot more thorough, but we still don't know exactly how this, repressing actually occurs. How does it work, right? And this is when scientists discovered actually 3 operator sites instead of just one. Ignore the CAP for now. Just see there's O1, O2, and O3, right? So one is very far upstream and one is pretty far downstream. And you can see their respective nucleotide sequences, and they're very largely conserved, like very much so, right? And scientists wanted to see, okay, if they were to put in mutations into these operators to basically make them not able to be bound by repressor, right? Sort of similar to our OC mutants that the scientists identified back then, right? That's denoted with the X's, meaning repressor can't bind there. Like how does that affect repression? Okay. And so obviously when you have all 3, everything's great. Things are looking good. When you remove one, just one, either 2 or 3, we have considerable decrease, right? But you can still, it still works, right? So you remove O2 and okay, it almost quarters, right? You get a quarter of the repression that you had before, or I guess a third. Okay, not great, but could be worse. Same with O3. You remove O3, you get about half as much as you had before. But as soon as you remove 2, look at this decrease. Huge decrease all the way down to 18. Compared to 1300, right? That's very, very low. And interestingly enough, if you remove O1, just O1, you get no repression whatsoever, basically. Essentially no repression. I mean, obviously after you remove more, it's not going to help, right? These are horrible. So we can get a few things from this. Firstly, we need at least one of the other 2 operator sequences, either O2 or O3, to get any meaningful repression. So that's hint number 1. So number 1, need at least O2 or O3. And 2, we need O1. These are the 2 biggest takeaways from this experiment. Because as soon as you remove O1, you lose all repression. As soon as you lose both O2 and O3, you lose all repression. You need at least O1 and one of the other 2. So keep this in mind. Now, X-ray crystallography revealed the structure of Lac repressor, and basically, it's a tetramer, right? So that means there's four proteins, 4 subunits. That has 2 dimers, and each dimer is made of 2 monomers that each can bind to a regulatory region. And since these are monomers, this is sort of why we see in the operator sequence, we can see these inverted or partially inverted sequences, because one monomer will bind here and the other monomer will bind here. Sort of what it looks like is this is one monomer, this is the other monomer, and the other place, this is one monomer, this is the other monomer. These 2 are the dimers, and then these dimers come together to form the repressor tetramer. Right, that's sort of the structure. That's what X-ray crystallography found. And so the proposed model for this is, it kind of follows, right? If we need at least O1 and another one, this sort of makes sense because, okay, well, one of these dimers will bind to O1, and the other one kind of needs to bind to something else too, because it will form what we see here is sort of like a ring structure. And obviously, what is RNA polymerase meant to do with this? It doesn't know what to do with this, right? And so we would have, let's say O1 is here, and then let's say this is O2, right? And RNA polymerase can't bind here. Same with if this is O3, right? RNA polymerase wouldn't be able to bind. However, if this, let's say, is O2 instead, right? Let's say it didn't bind to O1. Let's say O1 is down here. RNA polymerase can actually still bind. Because, I mean, the promoter, if we look at the map here, right? RNA polymerase binding site is right next to O1, right next to it. If our repressor is repressing at O3 and O2, forming this loop, the inside here is pretty intact, right? And clearly it must work because as soon as you break O1, we get no repression, meaning that we need binding at O1. 100%. We need at least one of the dimers to bind at O1, and the other one can be whatever, it can be either O3 or O2, and it'll work. So this sort of makes sense, right? This makes sense on a molecular level, and it agrees with all the experimental data we've found so far. Now, obviously, the next elephant in the room, right, is glucose. I mean, if we go all the way back to the start, right, our whole motivation was that glucose was used first, and then there was a switch from glucose to lactose. And that part, we solved, okay, we solved this half, great. We know why this happens, and we know what happens when there's no lactose. But we don't know why lactose isn't also used when glucose is used, right? That seems kind of odd. Like, why don't you just use both? So that's what we are going to look at next, is what's the role of glucose. Now, this might seem unrelated, but a key indicator of low glucose levels is this molecule called cyclic AMP. And so low glucose results in high cyclic AMP. And obviously we saw from the experiment that when you have a lot of glucose, right, you're not going to get a lot of, lactose metabolism, meaning not a lot of beta galactosidase, right? And so what they did was they treated cells, right, bacterial cells, with inducer, right, to remove repressor. But they also added glucose. So glucose, right, glucose should completely, inhibit this expression, right? As we saw in the experiment originally, right? Glucose is not going to let anything happen. So this is high glucose. But then they added CAMP as well, okay? They added CAMP on its own, right? Usually CAMP is naturally produced when there's low glucose. But when there's high glucose, it's not going to be produced, but these scientists added it anyway, and they saw that beta galactosidase was expressed after that. So this suggests that CAMP is a positive regulator, right? CAMP somehow activates or allows for the transcription of the Lac Operon. Obviously there was no repressor, so that works out, right? No repressor, so it was just the glucose that was the barrier. But as soon as you add a CAMP, there was no more barrier. So that suggests that glucose actually doesn't really do anything to the Lac Operon. In fact, it's CAMP instead. So that's what it says. Like, it can overcome glucose-mediated repression, right? You add CAMP, glucose doesn't matter anymore. So this suggests that it's just CAMP that is important. Now, they also found a catabolite activator protein. This is a protein they isolated that they thought might have played a role in this experiment. In this, they might have thought this played a role in this operon. So what they did is that they got some cell lysate, all right, and they added some Lac Operon DNA with no repressor gene, okay? So this means that this would be constitutive expression, right? There's no repressor to prevent binding, transcription. And they added CAMP and isolate and the CAP protein that we talked about, right? And they also found mutants that didn't grow on lactose, but they did have CAMP levels were high. Their CAMP levels were high, but they didn't grow on lactose. And so this kind of just suggests that there was some defect in what, probably the CAP gene, right? That they identified that they think might be some sort of activator, um, you know, for this operon. And so this was sort of the results they saw. So they plotted CAMP, cap protein, and beta gal, right? So CMP they either added some or they didn't. Same with cap protein, and then they measured the beta galactosidase activity. And okay, unsurprisingly enough, when you don't add either, right, there's no beta gal activity. Who would have guessed, right? When they added CMP but they didn't add cap protein, okay, they saw activity. Okay, that makes sense. Because the cells probably have CAP anyway. If it does play a role, right? You don't really like adding some. He's not going to help that much, but I mean, it kind of. If you look at this line, right? There's a little bit of increase in beta gal activity, so I guess it doesn't hurt, right? It doesn't hurt, but an important one is that this wild type, right? When you didn't add CAMP, there was no activity. You didn't see activity, so clearly you do need CAMP, which is kind of what we saw earlier. This agrees with our previous findings. Now, what's more interesting are these mutants. These mutants presumably have non-functional CAP protein. And that's exactly what we see here. Look, see, we add CMP, which we thought before was all we needed to activate expression of these genes. But it wasn't enough because there was no beta gal activity. We added CMP and we didn't get any activity. So what gives? Well, in this mutant, when we added CAP, we rescued some activity. It's not a lot, to be fair, but we did rescue some activity. And this activity was only rescued in the presence of CAMP. So this suggested that, okay, this CAP molecule is an activator for transcription. And this CAP is activated itself by CAMP. And without CAMP, this is non-functional. That's sort of like, the idea that this suggests. So obviously we're going to see isolate this protein for one, right? But also make sure that it does bind with CAMP. And this is the exact same radioactive assay that we did with the Lac Operon and IPTG, right? Exact same thing. And boom, we've isolated it. We know the size now, and that's great. But now they want to characterize this binding, right? Obviously, how does this bind to the sequence? Like that, now we got a cap protein now. Like, why is it getting so complicated? So they want to see how this worked. And they use DNA footprinting for this. And the way this works is you have a bunch of strands of DNA, right? That's all labeled on one side. And then you would add your protein of interest. Okay, let's say, so CAP, so it's going to be bound here, right? And then you add just enough DNAs, right? So DNAs will break DNA pieces into two, right? And just add enough so where it will make exactly one cut. So either cuts here, or it cuts here, or it cuts here, or it cuts here, right? But it wouldn't be able to cut where the protein is because, well, the protein is protecting it. And so when you're on a gel, you end up getting this DNA footprint, as you can see here. So obviously we have our controls. We have no protein, right? We have no protein. This is our negative control. Because, well, the DNA is just going to get cut anyway, right? And then we added CAP on its own with no CAMP, and okay, nothing changed. So this suggests that CAP does actually need CAMP to bind. Okay, that's great. This agrees with our model. We need CAMP for CAP to bind in the first place because these two lanes are identical. Now, when we added CAMP, we saw the footprint. Amazing. So it does exist, right? And it does bind. And very interestingly enough, there are these exposed DNA sequences that did get cleaved. And this suggests that there are some hypersensitive DNA bits that are shown by the CAP after they bind, right? This is even more interesting, right? Because it's not like we just see like a block, right? We actually see that there are some exposed DNA regions that, well, for one, they could be, accessed by the DNAs, so it must be outside. And this was very interesting because this would give hints as to how CAP, allowed for RNA polymerase binding, and facilitating its transcription. So Next up, we want to look at its regulation, right? So just to make sure, really solidify that this is in fact, um, it does in fact, just to make sure this is in fact involved with transcription. So what they did was they had DNA and they allowed RNA polymerase to bind, both in the presence and absence of CMP and CAP. And then you would add nucleotides and rifampicin, which would block transcription initiation. Meaning that, okay, if you're too late, you're too late. Like, you're too bad, right? And what they found was that you needed to add both CAMP and CAP for transcription. If you added it afterwards, right? Let's say you add your RNA polymerase, but nothing else, right? And let's say you had no repressor. There's no repressor. There's no, there's inducer, right? But RNA polymerase didn't manage to bind without CAMP and CAP. Because so first you add RNA polymerase, and then you add rifampicin, right? So this is going to block transcription initiation, and then you add CAMP, CAP. And maybe, okay, so these now bind to the DNA, but it's too late now. RNA polymerase can't transcribe anything because rifampicin will block transcription initiation. So, but as soon as you added it beforehand, we saw transcription, right? And so if we added it beforehand and we saw transcription, that would mean that, okay, CAMP and CAP are both required for RNA polymerase to bind in the first place and to transcribe. So, yeah, it suggests that CAP plays a big role in facilitating the formation of the open complex of RNA polymerase to allow it to transcribe. And that concludes our whole journey of the Lac Operon. And we can take a look at our final model now, because now we can bring it all together. So we started off by seeing that, okay, in the presence of glucose and lactose, there was this lag phase, right? This was very interesting because this suggests that there's some sort of switch that turned on the Lac Operon, right? Very, very interesting. And we've determined that there are 3 genes controlling this, right? Because these were all coupled to an inducer. And so let's call this the promoter. And then we had 3 genes, LAC-Z, LAC-Y, and LAC-A. LAC-Z is encodes for beta-galactosidase, right, which would break down lactose for metabolism. LAC-Y encodes for lactose permease, which brings in lactose into the cell to be used in the first place. And LAC-A encodes for transcellulase, which is required for this process, but its function is largely unknown. We also know that there's another gene called LAC-I that encodes for a tetramer protein of dimers that will repress the promoter. And this promoter region, we now know, has multiple sites. There's a P3, and we know that this repressor binds to these sequences called operators. And there are 3 of these operators. There's one right around the promoter. There's one a bit downstream, about -80 nucleotides downstream. And then there's one very far upstream at about like positive 400-ish. This is O3. It's O2. And this tetramer repressor will bind to O1, so there's one of the dimers. And then the other dimer will either bind to O2 or O3. And this will form a loop, like we see here, that prevents binding of RNA polymerase to the DNA. Now, an inducer, such as allolactose, will bind to this tetramer and release it from the DNA through conformational change. Maybe it'll bind up there, and then it releases it from the DNA, allowing for RNA polymerase to bind. But before we can let RNA polymerase to bind, we also know that there's this CAP protein, this catabolite activator protein, that must also bind to the operator, because this protein promotes the formation of the open complex RNAP, which is required. And now this protein requires CAMP, and CAMP is only produced in low lactose. So basically, if you want transcription of the Lac Operon, you need both low lactose or high CAMP, right? However you get that. But also you need presence of an inducer, which is usually lactose. So that's the only way you can get meaningful transcription of the Lac Operon, and this exactly explains what we see in this experiment, because we saw that glucose was used, right? This is normal. This is normal metabolism, right? This is the bacteria is using glucose, it's having a great time, and then it runs out. And as it runs out up here, we see CAMP levels rise. And obviously lactose was here, so lactose implies allolactose, right? And then allolactose will release the repressor. And so this is the perfect conditions for the Lac Operon to be expressed, and that's exactly what we see up here. And then obviously, the growth dies out after lactose is used. That is our final model for the Lac Operon, and we've gone through all the experiments, or not all the experiments, but we've gone through a lot of the experiments that have given evidence to support this model. And yeah, that's the Lac Operon for you.